So there are different phases of clinical trials. Once your new drug application is approved from clinical preclinical trials, now your drug is entered into clinical trials. Now you have to follow the stepwise phases of clinical trials. So phase first, that is nothing but the safety and tolerability. So in that you will have 10 to 100 participants. Usually the participants or the members or the subject must be healthy, and sometimes it may be patient with rare diseases. Okay. So what is the aim? Safety and tolerability, to know the maximum tolerable dose. Okay. Again, in the phase second, what is the main aim? That is therapeutic exploitation, to confirm the therapeutic efficacy and dose ranging establishment. Again, in phase second, there will be 50 to 500 patients, and these patients, they are receiving your experimental drug in this case. So randomized trials will be there. In phase 3, it will be multi-site trial. And what is the main aim of phase 3? That is a confirmation, therapeutic confirmation. Okay. So phase second is also known as therapeutic exploitation. Phase third is known as therapeutic confirmation. Okay. In the phase 3, you are confirming in the larger population that your drug is effective or not. In the phase 3, few hundreds to 1,000 patients will be there, and these GPAT 19 patients are receiving experimental drug. Phase 4, that is a post-marketing surveillance, and in that case, many patients, many thousands of patients or a larger population receiving the approved. In the phase 1, 2, 3, the drug is nothing but your experimental drug or investigational drug. But in the phase 4, your drug is approved. Patients are receiving approved drug in the phase 4. Remember this. So number of patients, that is also important. A lot of times they ask the question on this. And what is the main aim for phase 4? That is to determine the adverse drug reaction, compliance, then drug-drug interaction, etc. And in some books, phase 0 is also mentioned. That is a microdosing study, and subjects are healthy volunteers used. They are very small in numbers, 5 to 10. And the main aim of phase 0 is nothing but to know the pharmacokinetics of the drug. Is that clear? Now let's take a drug metabolism process. Am I audible to you clearly? So drug metabolism. So we have already completed your absorption of drug, distribution of drug, which is common in 3 subjects: pharmacology, then your medicinal chemistry, and biopharmaceutics. Okay, so all the kinetics, absorption, distribution, metabolism, elimination, all these are the same in the all these 3 subjects. So we are taking in a hybrid manner. Okay, so out of that, third kinetic step is nothing but the biotransformation. Suppose that you have taken a tablet orally, it is disintegrated, dissolution will be there in the stomach and GIT. After that, absorption will be there. After that, distribution will be there. Once it is distributed to various tissues and at the site of action, it will show its pharmacological action. What after that? अगर आपका ड्रग आपके पेट से एब्सॉर्ब होगा ब्लड में, उसके बाद ब्लड से डिस्ट्रीब्यूट होगा आपके जितने भी टिश्यूज है उसमें, और साइट ऑफ एक्शन में जहाँ पे उसको एक्ट करना है वहां पे भी गया, लेकिन उसके बाद क्या? वैसे तो नहीं रह सकता ना वो ड्रग बॉडी में. सो यू हैव टू, योर बॉडी हैज टू रिमूव दैट ड्रग आफ्टर इट्स पर्पस. जैसे ही उस ड्रग का पर्पस फुलफिल होता है, जैसे उसका काम होता है, जो काम के लिए आपने ड्रग लिया था वो काम हो गया, तो उसके बाद में भी वो ड्रग नहीं रह सकता बॉडी में. So it has to be removed from body, and you know that for absorption you need a unionized lipid soluble drug. Okay, unionized lipid soluble drug for absorption, and now for excretion you need a water soluble ionized drug. Okay, for the absorption you need a unionized lipid soluble molecule, and for the elimination you need a ionized water soluble molecule. And the process of making lipid soluble molecule into water soluble molecule, the process of making lipid soluble molecule into a water soluble molecule. ठीक है, आपको एब्सॉर्प्शन करने के लिए लिपिड सोल्यूबल लगता है, लेकिन बॉडी से रिमूव करने के लिए वाटर सोल्यूबल लगता है, क्योंकि मेजर रूट ऑफ रिमूवल इज यूरिन, राइट. तो आपको रिमूव करना है ड्रग बॉडी में से, तो उसको वाटर सोल्यूबल बनाना पड़ेगा. एंड द प्रोसेस ऑफ मेकिंग लिपिड सोल्यूबल ड्रग इनटू वाटर सोल्यूबल ड्रग, दैट प्रोसेस इज नथिंग बट द मेटाबॉलिज्म और बायोट्रांस. सो व्हाट इज बायोट्रांसफॉर्मेशन? सो बायोट्रांसफॉर्मेशन इज अ synonym for your metabolism. Biotransformation means transformation of active drug into inactive drug in the biological system. Okay, so your body is nothing but the biological system, and your body is transforming your active drug into inactive drug, your lipophilic drug into water soluble drug, so that it can be easily excreted from the body. That is nothing but the biotransformation. That is nothing but the metabolism. Okay, so it is nothing but the transformation of drug in a biological system, or conversion of drug from one form to another form, that is from active form to inactive form, or lipophilic to hydrophilic form, which may be either active or inactive. Okay, so we will see that. So biotransformation is transformation of drug in the biological system from one form to another form, which may be either active, inactive, more active. It will have other pharmacological activity. We will see that. Don't worry. So that is nothing but the that is nothing but the biotransformation. Okay. Now, what is the main aim of biotransformation? What is the main purpose of biotransformation? What is the main purpose of metabolism? So main aim of biotransformation is nothing but to make a drug water soluble. Okay. For absorption you need a lipid soluble drug. And for excretion you need a water soluble drug. ओके सो एब्सॉर्प्शन तो हो गया लिपिड सोल्यूबल ड्रग का, उसके बाद उसको एक्सक्रीट भी तो करना है, बॉडी में से बाहर तो निकलना है, उसके लिए वाटर सोल्यूबल बनाना पड़ेगा एंड द वाटर सोल्यूबल बनाने की प्रोसेस कौन करता है, ये बायो ट्रांसफॉर्मेशन। ओके सो मेन एम ऑफ बायो ट्रांसफॉर्मेशन इज टू मेक द ड्रग वाटर सोल्यूबल। इज दैट क्लियर? नाउ नेक्स्ट, ओके नाउ द मेटाबॉलिक फैक्ट्री ऑफ बॉडी, व्हाट इज मेटाबॉलिक फैक्ट्री ऑफ बॉडी? दैट इज नथिंग बट द लिवर, एंड दिस लिवर is used to metabolize all the drugs. Even most of the drugs are metabolized by liver. Okay, and drugs are nothing but the xenobiotics. Xenobiotics means, for xeno means foreign, biotics means our system. Those things which are foreign to our system, they are known as xenobiotics, or any foreign chemicals. So those chemicals, drugs are nothing but the xenobiotics. Okay, so metabolic factor of liver, body is nothing but the liver, and is which metabolize all the drugs and xenobiotics. Okay, the next point. So metabolism is the process. Okay, metabolism is the process of conversion. This line is very important. Metabolism is a process of conversion of unionized lipid soluble drug into ionized water soluble drug. Okay, metabolism is the process by which there is a conversion of unionized lipid soluble molecule to the ionized water soluble molecule so that it can be easily excreted. That's it. Okay, and this biotransformation is also known as detoxification process. This metabolism is also known as detoxification process. Okay, and after biotransformation you will get a water soluble drug, water soluble product, water soluble metabolite. Okay, that is the main aim of metabolism. So after biotransformation, drug result in the water soluble product. Okay, यहाँ पे now in this slide your 2 or 3 questions will be covered. So either they can ask intellectual question, either they can ask applied question, or either they can ask line to line. ऐसी इस तरीके से question पूछते हैं. Okay, so next point is drug metabolism result in. Okay, so what are the result of drug metabolism? कि आपका ड्रग मेटाबोलाइज हो गया बॉडी में, उसके बाद क्या होगा उसका, ठीक है? तो मेटाबोलिज्म होने के बाद क्या क्या बन सकता है, व्हाट आर द डिफरेंट पॉसिबिलिटीज, व्हाट आर द डिफरेंट आउटकम्स दैट विल कम इन फ्रंट ऑफ यू आफ्टर ड्रग मेटाबोलिज्म। कि अगर कोई ड्रग का मेटाबोलिज्म होता है, उसके बाद में उसका क्या होगा, उसके बाद में उसका नसीब में क्या है, उसके नसीब में 3 या 4 चीजें हैं, वी विल डिस्कस वन बाय वन। ओके, सो ड्रग मेटाबोलिज्म विल रिजल्ट इन आइदर फर्स्ट इनएक्टिवेशन ऑफ ड्रग। ओके, इनएक्टिवेशन ऑफ ड्रग मींस जो भी ड्रग आपने लिया है उसका इनएक्टिवेट हो जाएगा। ओके, सो मेटाबोलिज्म विल रिजल्ट इन आइदर इनएक्टिवेशन ऑफ ड्रग और जनरेशन ऑफ एक्टिव मेटाबोलाइट। ओके, फॉर एग्जांपल इफ यू आर यूजिंग प्रोड्रग्स, सो व्हाट इज प्रोड्रग? प्रोड्रग इज नथिंग बट दैट दीज आर द ड्रग व्हिच आर नॉट एक्टिव देमसेल्फ। ओके, एज इट इज दे आर नॉट एक्टिव। फॉर एग्जांपल levodopa, if you are using levodopa for Parkinsonism, it is a prodrug. Okay, as it is not effective, but once it is metabolized in the body, so levodopa will be metabolized in the body into dopamine. Now that dopamine is effective. Okay, so that is nothing but the prodrug. So prodrugs are the drugs which are not active themselves, but after conversion or after metabolism in the body they will become a active drug, right? Okay, so there will be either inactivation of drug, there will be generation of active metabolite, or there will be generation of active metabolite with different action. For example, I will explain. Diazepam मालूम है आपको? There is one drug known as diazepam. Okay, it is a sedative hypnotic. Okay, so that drug is converted into another active metabolite after the metabolism in the body. Diazepam is converted into 3 metabolites: temazepam, oxazepam, and temazepam, oxazepam, and there is another one metabolite. I am not remembering the name. So temazepam, oxazepam. Diazepam itself is a less potent. It is a sedative hypnotic. Okay, it has a sedative hypnotic property, but its metabolite, that is oxazepam, it has an anti-convergent activity. There is a change in activity also. So there will be inactivation of drug. There will be activation of inactive drug, that is a prodrug. There will be generation of active metabolite which is having other action. Another example I will use. I would like to give you another example. Epromide is the drug. Epromide is the drug which is antipsychotic drug, and after its conversion in the body, it is converted into your isoniazid, which is having antitubercular. Epromazide is having antipsychotic activity, and after epromazide का मेटाबॉलिज्म होता है बॉडी में. epromazide ऐसा एक ड्रग है जो साइकोसिस के लिए पागलपन के लिए यूज करते हैं. तो वो ड्रग जब लेते हैं लोग तो उसका मेटाबॉलिज्म होता है बॉडी में, तो उसका एक नया ड्रग बन जाता है मेटाबोलाइट, और वो नया मेटाबोलाइट होता है कौन सा, आइसोनियाजाइड. दैट इज नथिंग बट द वो एंटीट्यूबरकुलर ड्रग है. सो चेंज इन द एक्टिविटी आल्सो देयर. ओके, सो देयर मेटाबॉलिज्म विल रिजल्ट इन इनएक्टिवेशन ऑफ ड्रग, then generation of active metabolite from prodrug, and or generation of active metabolite from active drug also, and from generation of active metabolite which is having another action from parent drug. Then second, what will happen occasionally? So this activation of drug, inactivation of drug, generation of active metabolite, and generation of active metabolite, this happens normally, okay, after metabolism. Okay, but occasionally what happens, activation of pharmacologically inactive drug like prodrugs. I already explained about this. So metabolism will result in inactivation of drug, generation of active metabolite which is having different action, then generation of your pharmacologically active drug, occasionally activation of pharmacologically inactive drug into active drug, that is a prodrug. I have given the example of levodopa. Levodopa as such is not, but once levodopa is given, it will go in the body and it will metabolize, and it will be converted into dopamine, and that dopamine is effective. So that is levodopa is nothing but the prodrug. Okay, so inactivation of drug, अगर आपने पैरासिटामोल लिया, पैरासिटामोल खाया, उसका एब्सॉर्प्शन डिस्ट्रीब्यूशन हो गया, एक्शन हो गया, अब मेटाबॉलिज्म होगा उसका, और वो पैरासिटामोल रिमूव कर दिया जाएगा बाहर, तो उसको बोलते हैं इनएक्टिवेशन ऑफ ड्रग. नाउ जनरेशन ऑफ एक्टिव मेटाबोलाइट, आपने डायजिबम लिया, डायजिबम आपके बॉडी में एब्सॉर्ब हो गया, डिस्ट्रीब्यूट हो गया, मेटाबोलाइज हो गया, मेटाबोलाइज होने के बाद उसका मेटाबोलाइट बन गया टेमाजेपम. सो दैट डायजिबम itself is active. Diazepam itself is an active drug, and it is converted into temazepam in the body. That temazepam is also active. So generation of active metabolite from either active drug or inactive drug. अगर if it is inactive drug, that is called, we call it as a prodrug. Okay, अगर वो inactive drug से active बनता है तो उसको हम prodrug बोलेंगे. Active drug से active बनता है तो उसको अपन active metabolite बोलेंगे. तो biotransformation or metabolism will result in inactivation of drug, generation of active metabolite of active drug, then generation of active metabolite of active drug which is having different activity. मैंने जैसे ऑक्सीजेपम का एग्जांपल दिया था, जैसे इप्रोमियाजाइड का एग्जांपल दिया. There is other action. We will take it again. Don't worry. वहाँ पे चार्ट है मेरा, वो चार्ट डिस्कस. So occasionally it will also result in the pharmacologically activation of pharmacologically inactive drug into active drug. That is a prodrug. Activation of prodrug. So metabolism will result in inactivation, activation, then toxicological activation. So rarely it will also result in the toxicological activation. That means generation of more toxic metal. So this happens with the few drugs. For example, जैसे आपका organophosphorus poisoning होता है, pesticides होता है. Pesticides after metabolism they become more toxic. Pesticides itself they are toxic, but after metabolism in the body they become more toxic. So there is a generation of more toxic metabolite of already toxic. Okay, so metabolism of drug will either result in inactivation of drug, generation of active metabolite of active drug, generation of active metabolite of active drug having different activity, then occasionally it will result in the activation of pharmacologically inactive drug, that is prodrug, then rarely it will result in the toxicological activation, that is generation of metabolite having more toxicity. Is that clear? These are the possibilities. Okay, ये पॉसिबिलिटी है मेटाबॉलिज्म के बाद क्या होगा उसका, ये पॉसिबिलिटी है. आइदर उसका इनएक्टिवेशन हो जाएगा, आइदर उसका एक्टिव मेटाबोलाइट बनेगा, आइदर उसका एक्टिव मेटाबोलाइट बनेगा कि जो दूसरी एक्शन दिखाए, या फिर और क्या होगा, ऑकेजनली ऐसा भी हो सकता है कि फार्माकोलॉजिकली इनएक्टिव ड्रग है, प्रोड्रग है, उसका एक्टिवेशन हो जाएगा. ठीक है, और ये भी हो सकता है कि जो टॉक्सिक ड्रग है उसका मोर टॉक्सिक मेटाबोलाइट. सो दीज आर द पॉसिबिलिटी ऑफ drug metabolism. Now we'll see the next. Okay, I have already explained these two terms in the very first lecture of our course. What do you mean by soft drugs and what do you mean by hard drugs? So those drugs which are easy to metabolize in the body, those drugs which will undergo metabolism easily in the body, those are known as soft drugs. And those drugs which are relatively resist the metabolism, those drugs which are not easy to metabolize, they are known as hard drugs. And I also have already explained these terms with examples in the very first lecture. Again, different organs of metabolism. and their order of ability to metabolize the drug. So there are different there are different organs which will metabolize the drugs. Okay, so there are liver. There is a main organ which will metabolize your 90% of drugs. Then again lungs are there. So few drugs are also metabolized in the lung. Then kidney is there. Then adrenal gland is there. Then testes are there. Skin is there. There are different organs which can metabolize the, but mostly drugs are metabolized by liver itself, because liver is a metabolic factory of body. That clear? Now see here this line. So most of the drugs will be metabolized by liver. Okay. So liver has a highest capacity to metabolize the drugs. After liver, lungs has a capacity. After lungs, kidney has a capacity. After kidney, intestine has capacity. After that, placenta. After that, adrenal. And after that, skin. Right? So they may ask you the question, which of the following organ has the highest capacity to metabolize the drug? So in that case, your answer should be liver. And I think they have already asked question related to this. Okay. सबसे ज्यादा कैपेसिटी होता है लिवर का, उसके बाद लंग्स का, उसके बाद किडनी का, उसके बाद इंटेस्टाइन का, उसके बाद प्लासेंटा, उसके बाद एड्रेनल, उसके बाद स्किन. सबसे कम मेटाबोलाइज होता है ड्रग स्किन में, सबसे ज्यादा मेटाबोलाइज होता है लिवर में. ये रिपीट क्यों कर रहा हूं मैं आपको, इसके पीछे रीजन है, क्योंकि ये क्वेश्चंस आते हैं, ये मैंने देखा है. सो नेक्स्ट, so drug metabolizing enzymes. Now what are the enzymes that are metabolizing the drugs? So there are enzymes, those are metabolizing the drugs. So they may be either microsomal enzymes or non-microsomal enzymes. So there are two types of enzyme which metabolizes the drugs. दो तरीके के एंजाइम होते हैं जो मेटाबॉलिज्म में इन्वॉल्व होते हैं. फर्स्ट वन इज अ माइक्रोसोमल एंजाइम्स एंड second one is a non-microsomal enzyme. So mostly microsomal enzymes will metabolize the drug. That doesn't mean non-microsomal नहीं करेगा, दोनों करेगी, लेकिन मोस्टली कौन करता है? माइक्रोसोमल एंजाइम. लेकिन माइक्रोसोमल एंजाइम्स होते क्या है? What are the microsomal enzymes? Now I will explain this. See, so your liver cell has a Kupper cell. Liver cell has a specialized cell for metabolism known as a Kupper cell. And in that Kupper cell, जो भी सेल का जो आपका endoplasmic reticulum होता है, so there is endoplasmic reticulum in the cells, and there are two types: smooth endoplasmic reticulum and rough endoplasmic reticulum. So particularly smooth endoplasmic reticulum जो होता है, जो सेल्स का, लिवर के सेल्स का, smooth endoplasmic reticulum of liver cell, they have microsomes. Microsomes means they are a spherical structures. They are a spherical structure in which the enzymes are stored. Okay, and whenever there is requirement, the enzymes are released from that microsomes, and that's why they are originating from microsomes, and that's why they are known as a microsomal enzymes. Okay, so microsomal enzymes, they are mostly present in the liver. Is that clear? Non-microsomal enzymes means enzymes those which are not those which are present in the other organs like intestine. जैसे आपके स्टमक में होता है कुछ enzymes, intestine में होता है, liver lungs में होता है, kidney में होता है. So all those are the non-microsomal enzymes. Okay, so most of the drugs are metabolized by microsomal enzymes. Is that clear? Now see this table. Effect of biotransformation on the drugs. Okay, जो पहले इसके पहले स्लाइड में मैंने बोला था कि what are the possibilities of drug, what will happen after metabolism with the drug. So again we will repeat it. Okay, so effect of biotransformation on the drugs. So what will be the impact, what will be the effect of metabolism on the drug? So first, there will be either inactivation of active, there will be either inactivation. Main aim of metabolism is nothing but the inactivation. So inactivation of active drug. For example, chloramphenicol is an antibiotic, is inactivated by conjugation. Okay, it is inactivated by conjugation reaction, and also along with other drugs, inactivation of drug like chloramphenicol. Then activation of drug to the active product. Okay, activation of drug of inactive drug. So prodrug. For example, in that case also there are two types which are therapeutically useful, which are toxic. So therapeutically useful, for example, levodopa. Levodopa is a prodrug and it is converted into the body after metabolism in the dopamine. And dopamine is useful. So dopamine is active, levodopa is inactive. But why we are making levodopa? Why we are giving levodopa? We can give dopamine also. There is a reason. Dopamine is a monoamine, right? It's a monoamine. Okay. And it is destroyed in our GIT. So you cannot use dopamine as it is. So you have to use levodopa. So levodopa is not destroyed in our gut. It is not destroyed in our blood. Dopamine is destroyed in the gut as well as blood. So it is a biogenic amine. So that's why we have to use prodrugs sometimes also. So activation of inactive drug, that is activation of lead over to dopamine. Then again April to enaprylate, generation of active metabolite, April to enaprylate. Then generation of toxic metabolite, for example parathion. Parathion is a pesticide. Okay, so after metabolism parathion is converted into peroxone. Parathion is converted into peroxone. Peroxone is a 20 times more toxic than this parathion. So generation of toxic metabolite which is more toxic than parathion, right? Then generation of active metabolite of active drug. Okay, for example इसमें भी दो होता है, more active. So if diazepam is given, it will be converted into oxazepam in the body, and this oxazepam is more active than diazepam. So generation of active metabolite of active drug which is a more active. Okay, now having different activity. Generation of active drug from active drug, generation of active metabolite from active drug which is having different activity. So here is one drug pethidine. It is used in the CNS depression. It is a narcotic drug. So it is a CNS depressant drug. After metabolism, it is converted into norpethidine. Norpethidine is a CNS stimulant. Pethidine originally it is a CNS depressant, but norpethidine is a CNS stimulant. Is generation of active drug into active metabolite having different action. So previous slide में मैंने आपको दूसरा example दिए थे, यहाँ पे दूसरा example. Now next, so metabolism of drug. Okay, it is carried out in the liver. Liver is the main organ to metabolize the drugs, and the metabolism of drug is carried out into different steps. Okay, so different phases. So phase first and phase second. There are different phases for the drug metabolism. Phase first, phase second. Okay, so phase first is known as, some books also mention, in the some books they have also mentioned phase 3, but for you it is not important. Just remember phase 1, phase 2. So in the phase 1 is write down in the paper, in your notebook. Phase first is also known as functionalization reactions. Phase first is known as functionalization reactions, and phase second is known as synthetic reactions or conjugated reactions or conjugation reactions. Phase first is a functionalization reaction, phase second is a synthetic reactions or conjugation reaction. जो भी ड्रग का मेटाबॉलिज्म होता है वो किस में होता है, ये दो फेजेस में होता है. जो फेज फर्स्ट होता है वो फंक्शनलाइजेशन रिएक्शंस होता है, फेज सेकंड होता है वो सिंथेटिक रिएक्शंस होता है और कंजुगेशन रिएक्शंस होता है. फेज फर्स्ट में क्या आता है, फेज फर्स्ट में क्या होता है, फेज फर्स्ट में होता है आपका ऑक्सीडेशन, रिडक्शन, हाइड्रोलिसिस. जो भी मॉलिक्यूल है उसका ऑक्सीडेशन हो जाएगा, इधर उसका रिडक्शन हो जाएगा, इधर उसका हाइड्रोलिसिस हो जाएगा. अगर वो अल्कोहल है तो उसका हाइड्रोलिसिस हो जाएगा, अगर वो ईथर है तो उसका हाइड्रोलिसिस हो जाएगा, अगर वो एसिड है तो उसका हाइड्रोलिसिस हो जाएगा. लाइक ऑक्सीडेशन हो जाएगा, मतलब डायजेपम इज कन्वर्टेड इनटू ऑक्सीजेपम बाय ऑक्सीडेशन. रिडक्शन में क्या होगा, रिडक्शन मतलब देयर विल बी रिडक्शन ऑफ दैट मॉलिक्यूल. फॉर एग्जांपल sulfur diazine, there is one drug known as sulfur diazine. Sulfur diazine is an inactive drug and it is used in treatment of diarrhea. ठीक है, तो ये ड्रग जब देते हैं तो इंटेस्टाइन में जाता है. इंटेस्टाइन में आपके बैक्टीरिया होते हैं known as azobacteria. So azoreductase bacteria. So they used to secrete one enzyme azoreductase. So that azoreductase reduce your sulfasalazine into two molecules and that will act local. So oxidation, reduction and hydrolysis. These three are the phase first reaction. Phase second reaction, they are known as a conjugation reaction or synthetic reaction. Phase first is nothing but the functionalization. So what happens in the phase first? There is a incorporation, there is addition of groups. In the phase first, there is addition of hydroxyl group, there is addition of carboxyl group, there is addition of nitro group, there is addition of amino group, like that. So these are the addition reactions, oxidation reaction, hydrolysis. What is the main aim of these reactions? Phase first में क्या होता है, ये जो groups होते हैं polar groups, so hydroxyl group, nitro groups, alcohol groups, all these are the polar groups. So phase first में क्या होता है कि आपका जो ड्रग है उसके ऊपर polar group का एडिशन होता है, ताकि वो ड्रग क्यों कर रहे हैं सब, क्योंकि वो ड्रग नॉन पोलर है उसको पोलर बनने का है. ठीक है ना, तो ये सब एफर्ट इसके लिए चल रहा है कि पोलर बनने का उस ड्रग को. सो देयर विल बी इंट्रोडक्शन ऑफ पोलर ग्रुप्स ऑन द फेज फर्स्ट एंड फेज सेकंड. क्या अगर वो उसमें भी पोलर नहीं बन गया तो फेज सेकंड में क्या होता है, उसको कॉन्जुगेट करते हैं. डिफरेंट कॉन्जुगेशन के लिए होता है अपना ग्लूकोनाइड कॉन्जुगेशन, सल्फेट कॉन्जुगेशन, ग्लूटाथायोन कॉन्जुगेशंस, देन एस्टाइलेशन. We'll see it one by one. So in the phase second there is a conjugation reaction. So in that also there are different reactions. First one is a glucourinide conjugation. Okay, and remember, write down, this is the most common phase second reaction. Glucourinide conjugation is the most common phase of phase second metabolism reaction, and this was question in GPAT. So most common phase second reaction in the drug metabolism is, and your options were there: glucourinide conjugation, glutathione conjugation, sulfate conjugation. I don't know the year exactly, but there was a question, glucourinide conjugation. This is the most common phase second reaction, right? Now next, acetylation. So acetylation means what? There is a introduction of acetyl group on the your molecule. Okay, so that is known as acetylation. Okay, so now different races of human beings will have different capacity to acetylate. So that's why depending on that there are slow acetylators or rapid acetylators, and due to this genetic difference of acetylation from person to person, this is known as genetic polymorphism, and this was the question in GPAT 20. Genetic polymorphism occurs with which kind of reaction? And there was a methylation, there was a conjugation, there was oxidation, acetylation, and the answer was acetylation. So genetic polymorphism is there in case of acetylation reactions. That is a phase second reaction. So multiple gene will control the enzyme N-acetyl transferase in different subjects or different races. कि अमेरिकन्स होंगे जो जल्दी एसिटाइलेशन रिएक्शन होगा उनमें, जो इंडियंस होंगे उनमें स्लोली एसिटाइलेशन रिएक्शन. दैट इज नोन एज रेशियल पॉलीमॉर्फिज्म और जेनेटिक पॉलीमॉर्फिज्म. ओके, एंड सब्जेक्ट्स कैन बी कंसिडर्ड एज either slow acetylators or fast acetylators. I will explain. I have examples. I have one example over here. I will explain. In case of slow acetylators, there will be slow acetylation reaction. In case of rapid acetylators, there will be quick acetylation reaction. Okay, so now see, there is one drug known as a anti-tubercular drug. There is one drug known as a isoniazid. Isoniazid is a drug used in tuberculosis. Okay, it is a first line agent used in tuberculosis, and different races, different people from different country will acetylize this isoniazid in different manner. कि अमेरिकन पीपल है वो जल्दी करेंगे एसिटाइलेशन उसका, या इंडियन पीपल थोड़ा सा स्लोली करेंगे. वो जो डिफरेंस होता है, दैट इज द डिफरेंस इन जेनेटिक polymorphism. So that is known as genetic polymorphism. So the capacity of different races to metabolize the drug will be different, particularly for the acetylation reaction, and that difference is known as genetic polymorphism. This question was asked in GPAT 20. See, isoniazid has different adverse effect. Isoniazid has a peripheral neuropathy as adverse effect. Isoniazid has a hepatic necrosis as adverse effect. Now what happens? Peripheral neuropathy is those who are slow acetylators of your isoniazid. Those patients, those people in which there is a slow acetylation of this isoniazid, they will have adverse effect of that is peripheral neuropathy. And those people who are fast acetylators or rapid acetylators of isoniazid, they will have a hepatic necrosis as adverse effect. Because जैसे ही फास्ट एसिटाइलेशन होता है वैसे ही फास्ट उसका मेटाबोलाइट बनता है, और वो मेटाबोलाइट वो लिवर को डैमेज करता है. जो स्लो एसिटाइलेटर्स होते हैं उसमें वो मेटाबोलाइट ज्यादा बनता नहीं, उनको वहाँ पे पेरिफेरल न्यूरोपैथी का एडवर्स इफेक्ट. Again, about this acetylators, which one is a slow acetylator, which one is a fast or rapid acetylators, we have one table in the next slide. We will discuss in that. Now here next point is methylation, sulfate conjugation, glycine conjugation, glutathione conjugation are the other phase second reaction. Okay, so phase first reaction oxidation reduction hydrolysis, phase second reaction conjugation reaction, that is glucuronide conjugation, acetylation, methylation, sulfate conjugation, glycine conjugation, glutathione conjugation and other. All these are the phase second reaction. Remember, ये इसमें एक बार क्वेश्चन ऐसा भी आया था कि उन्होंने एक रिएक्शन के नाम दिए थे, which one is not a phase second reaction, so which one is not a phase one reaction. So they can ask question in this manner. You must be clear about which are the phase first reaction, phase second reaction of metabolism, right? Now we'll see the next point. Molecular rearrangement involved in the metabolism of atracurium is a hormone. So atracurium is a drug. It is a skeletal muscle relaxant drug, atracurium. Okay. So what happens in the metabolism of atracurium? There is a Hoffman elimination. Hoffman elimination is there. Just remember the name. Hoffman elimination is involved in the metabolism of atracurium. एक बार पूछा गया था, ये वन लाइनर क्वेश्चन है, इसको सिर्फ याद रखिए, ज्यादा कुछ डिटेल में जाने की जरूरत नहीं. नाउ मोस्ट ऑफ द ड्रग्स आर मेटाबोलाइज्ड बाय माइक्रोसोमल एंजाइम P450 family. So most of the drugs, they are metabolized by microsomal enzymes, that is a P450 family, and they are given the name P450 because they will absorb UV radiations at the wavelength of 450 nanometer. just. So most of the drugs are metabolized by your microsomal enzyme P4, and that P450 is nothing but the why it is P450 given the name, that these enzymes have a lambda max at 450 nanometers. They will have absorption maxima at 450 nanometers. That's why they have given the P450. And they have different. There are different family of these enzymes. There are different isoenzymes, and on that also they used to ask the question. We will take that in the next slide. So yes, this is ethnic variation in the acetylation of isoniazide. So जो previously मैंने बोला था acetylation में slow acetylators, rapid acetylators. So white people जो गोरे लोग होते हैं, गोरे लोग है यूएसए के, कनाडा के, उसमें 45% स्लो एस्टाइलेटर्स है, उसमें 55% रैपिड एस्टाइलेटर्स. जो काले लोग होते हैं यूएसए में, black people are there in USA. These are 48% slow acetylators and 2% rapid acetylators. No need to remember the values and all that. Just be clear your concept. So Latin Americans होते हैं, अपने एक ही ड्रग है आइसोनियाजाइड, उसका मेटाबॉलिज्म डिफरेंट पीपल में कैसे होता है वो देख रहे हैं. So आइसोनियाजाइड is anti-tubercular drug and it is metabolized by acetylation reaction, and different people will have different rate of acetylation. So depending on that, people are classified, either they are slow acetylators or either they are rapid acetylators. Okay, so white people in the USA, they have 45% slow, 55% rapid acetylators. Black people in USA, 48% slow acetylators and 52% rapid acetylators. Japanese are slow acetylators. 87% of Japanese people, they are slow acetylators. 95% of Eskimos, 95% of Eskimos. Eskimos means peoples of Canada, Greenland and Russia. So they are 95% slow acetylators. And 100% of Orientals. Orientals means peoples of East and Southeast Asia. Peoples of East and Southeast Asia, particularly Chinese people, then Chinese people and the Russian peoples. These are the Oriental peoples and group of Oriental. उनका जात का नाम होता है Oriental. Eskimos, उनका ट्राइबल का नाम होता है. So they are the 100% rapid acetylators. It was just a examples. Now we'll see. Most of the drugs are metabolized by CYP3A4/5. Most of the drugs are metabolized by microsomal enzyme, that is cytochrome P450 3A4/5. Remember this line. Most of the drugs are metabolized by this enzyme CYP3A4, and this was the question in GPAT 2000. So most of the drugs are metabolized by this enzyme. Always this is not a case. सब में यही एंजाइम मेटाबोलाइज करेगा ऐसा कहीं जरूरी नहीं. बट एज अ नॉर्मली सबसे ज्यादा मेटाबोलिज्म जो होता है साइटोक्रोम, मतलब जो भी लीवर में वो ये एंजाइम करता है CYP3A. उसके सब टाइप्स भी होते हैं साइटोक्रोम के एंजाइम के. तो क्या होता है microsomal enzymes and their, here you have a table. Okay, cytochrome P450 enzymes and their isoenzymes. Okay, first one is a CYP3A4/5. 36% of drugs, they are metabolized by only this enzyme. So only one enzyme that will metabolize 36% of drugs, that is known as a CYP3A4/5. And this was question in GPAT 2000. Then after that CYP2D6 that will metabolize 19%. अगर आपने 100% कंसिडर किया कि 100% ड्रग्स है, उसमें का कितना परसेंट कौन मेटाबोलाइज करेगा, तो 36% ये वाला एंजाइम मेटाबोलाइज करेगा CYP3A4/5. उसके बाद आता है आपका CYP2D6, वो 19% मेटाबोलाइज करेगा. उसके बाद CYP2C9/9, उसके बाद ये 16% करेगा. उसके बाद आता है CYP1A2, वो 11% करेगा. उसके बाद CYP2C19, ये 8% करेगा. CYP2E1 4%, CYP2A6 3%, CYP2B6 3%. So most of the drugs, they are metabolized by this enzyme 4/5. Remember this line. Again, what are the enzyme inducers? What are the enzyme inhibitors? Okay, so enzyme inducers are nothing but those drugs which will induce the enzyme, which will increase the functioning of enzyme, which will increase the functioning of enzyme. They will induce. वो एक हिंदी में कहावत होती है, आ बैल मुझे मार. तो आ बैल मुझे मार मतलब क्या, कि एक ड्रग है, उसका मेटाबॉलिज्म होने वाला है बॉडी में, तो वो क्या कर रहा है, वो एंजाइम को बोल रहा है आ बैल मुझे मार. सो those are the enzymes. Those will accelerate the metabolism of themselves. Few drugs are the drugs those will induce the enzymes and they will accelerate their own metabolism along with other drugs taken with them. So that is known as enzyme inducer. And what are the enzyme inhibitors? These are the drugs which will inhibit or which will decrease the functioning of enzymes. Okay, enzyme inducers are the drugs those will increase the functioning of your enzymes, your metabolizing enzyme. Enzyme inhibitors are the drugs those drugs which will decrease the functioning of metabolizing enzyme. So enzyme inducer अगर है तो वो क्या करेगा, उसका मेटाबॉलिज्म खुद ही जल्दी करेगा, या फिर उसके साथ में अगर कोई दूसरा ड्रग दिया है तो उसका भी मेटाबॉलिज्म कर देगा। तो एंजाइम इंड्यूसर। एंजाइम इनहिबिटर्स क्या होता है? अगर एंजाइम इनहिबिटर्स क्या होता है, वो ड्रग अगर लिया आपने, उसका मेटाबॉलिज्म इनहिबिट करेगा, तो जल्दी नहीं होगा उसका मेटाबॉलिज्म। और उसके साथ में अगर कोई ड्रग लिया तो उसका भी मेटाबॉलिज्म वो कम कर देगा। तो उसके हिसाब से एंजाइम इंड्यूसर एंड इनहिबिटर्स यू हैव टू टेक केयर कि एंजाइम इंड्यूसर है तो उसका करंट में दूसरा ड्रग है तो उसका क्या होगा, उसका मेटाबॉलिज्म लो होगा। अगर एंजाइम इनहिबिटर है ये ड्रग, उसके साथ में जो दूसरा ड्रग दिया तो उसका मेटाबॉलिज्म कम होगा। लाइक दैट interaction will be there. So we have charts of a drug which those are the drug which will increase the metabolism. So drug induced acceleration or drug induced metabolism of drug. So barbiturates अगर आपको दिया है, so if you are taking the barbiturates or if you have been prescribed barbiturates for certain conditions, these barbiturates are enzyme inducers. They are the enzyme inducers. That means they will induce enzyme and they will accelerate, they will increase, they will speed up the metabolism of themselves as well as drug given along with them, right? So barbiturates क्या करेंगे, वो खुद का तो मेटाबॉलिज्म इंक्रीज करेंगे, लेकिन उनके साथ में जो अदर ड्रग, उसके साथ में अगर कोई ड्रग दे तो उसका भी मेटाबॉलिज्म इंक्रीज करेंगे। तो बार्बिट्रेट के साथ में अगर ये देंगे कौन सा, एंटीकोआगुलेंट, स्टेरॉइड्स, कॉन्ट्रासेप्टिव्स, विटामिन्स, पैराक्सिन, फिनाइलबिटाजोन, पेंटाइन, ग्रिजियोफुल्विन, कोलमफेनिकोल, थियोफाइलिन, ये चार्ट आपके बुक में भी है, ठीक है? तो जस्ट एक रिवीजन के लिए आप ले रहे हैं। तो इनका मेटाबॉलिज्म बार्बिट्रेट एक्सेलरेट करेगा। जैसे फिनेटोइन है, एंटी एपिलेप्टिक ड्रग, ये खुद का मेटाबॉलिज्म भी इंक्रीज कर देगा और उसके साथ में ये ड्रग का भी मेटाबॉलिज्म इंक्रीज कर देगा। तो इनका बायो अवेलेबिलिटी कम रहेगा। अगर ज्यादा मेटाबॉलिज्म होगा क्विकली तो बायो अवेलेबिलिटी ऑब्वियसली कम रहेगा। रिपम्पिन will increase the metabolism of these drugs. Contraceptives, glucocorticoids, then propranolol, metoprolol. Carbamazepine will increase the metabolism of vitamin D. So diclofan and gamma benzene hydrochloride, they are powerful microsomal enzyme inducer. Okay, so this gamma benzene hydrochloride, it is used in the treatment of scabies. अगर आपको खरोच हो गया, खुजली हो गया, उसमें वो ट्रीटमेंट में यूज करते हैं गामा बेंजीन हाइड्रोक्लोराइड, उसका इमल्शन आता है, ठीक है? तो दीज ड्रग्स आर पावरफुल एंजाइम इंड्यूसर्स। अगेन निकोटीन इज आल्सो पावरफुल एंजाइम इंड्यूसर। अगर आप सिगरेट पीने का शौक रखते हो तो निकोटीन जो है सिगरेट में का, निकोटीन इज आल्सो enzyme inducer. Okay, this was the question in GPAT also. Cigarette smoking will also induce the enzyme and it will also increase the metabolism of certain drugs. जो सिगरेट पीते हैं, जिसको सिगरेट पीने की लत है, उनमें मेटाबॉलिज्म जल्दी होता है। क्यों? क्योंकि निकोटीन इज अ एंजाइम इंड्यूसर। दैट क्लियर? जो सिगरेट पीते हैं उनमें ड्रग का मेटाबॉलिज्म जल्दी हो जाए क्योंकि इंड्यूसर है। नो, लेट्स सी द सेकंड चार्ट। ड्रग इंड्यूस्ड इनहिबिशन ऑफ मेटाबॉलिज्म। ये ऐसे कौन से ड्रग है कि जो मेटाबॉलिज्म को कम करेंगे, ओके, या इनहिबिट करेंगे? सो हियर आर द ड्रग्स। एलोपिरिनॉल इज देयर। एलोपिरिनॉल विल डिक्रीज द मेटाबॉलिज्म ऑफ these drugs. Tolbutamide, methotrexate, probenecid. Disulfiram is a drug used in the treatment of alcohol intoxication या अल्कोहल की लत छुड़ाने के लिए डाइसल्फिराम नाम का ड्रग यूज किया जाता है। दैट इज नथिंग बट द अल्कोहल डिहाइड्रोजनेज इनहिबिटर एंड इट विल decrease the metabolism of alcohol, tolbutamide, warfarin. Isoniazid will decrease the metabolism of these carbamazepine. Chlorpheniramine will decrease the metabolism of tolbutamide, probenecid. ये सिर्फ एग्जांपल है, आपको ज्यादा कुछ किए करने की जरूरत नहीं। जब आपको सब कुछ हो जाएगा तब आपको समझ में आएगा कि ये ग्लूकोकॉर्टिकॉइड्स क्या है, कॉन्ट्रासेप्टिव्स क्या है, कार्बामेजेपिन क्या है, प्रोबेनिसिड क्या है, वारफेरिन क्या है, डायजेपिन क्या है। अभी सिर्फ इतना समझ में आना चाहिए कि कुछ ड्रग ऐसे होते हैं कि जो एंजाइम मेटाबॉलिज्म को इंक्रीज करते हैं। अगर वो इंक्रीज करते हैं मेटाबॉलिज्म, मतलब एंजाइम इंड्यूसर है, तो वो क्या करेंगे, वो खुद का मेटाबॉलिज्म भी इंक्रीज कर देंगे, लेकिन उसके साथ में अगर दूसरा कोई ड्रग लिया तो उसका मेटाबॉलिज्म भी इंक्रीज। और कुछ ऐसे ड्रग्स होते हैं कि दे आर एंजाइम इनहिबिटर्स और दे आर एंजाइम इनहिबिटर्स, दे आर इनहिबिट द मेटाबॉलिज्म, तो वो उनका खुद का मेटाबॉलिज्म भी कम कर देंगे और साथ में जो ड्रग दिया उनका भी कम कर देंगे। अभी जो ड्रग मेटाबॉलिज्म बढ़ाते हैं वो दूसरे ड्रग का बायो अवेलेबिलिटी कम करते हैं, उसका इफेक्ट कम। और जो ड्रग एंजाइम जो ड्रग मेटाबॉलिज्म को कम करते हैं, इनिबिट करते हैं, वो क्या कर रहे हैं, वो दूसरे ड्रग का एक्टिविटी बढ़ा रहे हैं। इन द सेंस समझ में आया कि आपको, अगर मान लीजिए कि आपने 2 ड्रग लिया, ड्रग पैरासिटामोल लिया और आपने उसके साथ में अगर कोई एंजाइम इंड्यूसर ड्रग लिया, तो वो एंजाइम इंड्यूसर ड्रग क्या करेगा, एंजाइम को इंड्यूस करेगा मेटाबॉलिज्म के लिए, उसको बुलाएगा आबाइल मुझे मार। तो फिर पैरासिटामोल का नॉर्मली उसका जो टाइम स्पेन है वो 6 आवर्स है। 6 आवर्स में ब्लड में रहता है पैरासिटामोल। तो अगर एंजाइम इंड्यूसर है उसके साथ में, तो 6 आवर्स के बदले 3 आवर्स में ही उसका मेटाबॉलिज्म हो जाएगा, पैरासिटामोल का इफेक्टिवनेस कम हो जाएगा। सो एंजाइम इंड्यूसर विल अफेक्ट द effectiveness of other drugs. Enzyme inducers will also affect the metabolism. Enzyme inducers will affect the activity of other drug. Again, enzyme inhibitors will also affect the activity of other drug. So, enzyme inhibitors will increase the activity of other drug. Enzyme inducers will decrease the activity of other drugs. Clear? ये concept clear कर लो, बाकी कुछ नहीं। ये एग्जाम्पल्स आप बाद में भी पढ़ सकते हो, बाद में रट्टा मारो, कुछ क्या करना है आप देखो, ठीक है। ये रट्टा मारने की जरूरत नहीं, उसको 1-2 बार भी पढ़ लिया तो याद रहेगा। तो दीज आर विक्टिम ड्रग्स, मतलब यहाँ पे लिखा है इनिबिटर और विक्टिम ड्रग। इसका मतलब क्या है? इनिबिटर मतलब ये एलोपिरिनॉल विल इनिबिट द मेटाबॉलिज्म। किसका मेटाबॉलिज्म इनिबिट करेगा? ये विक्टिम ड्रग्स है, ये ड्रग विक्टिम है। अगर ये ड्रग साथ में दिए एलोपिरिनॉल और ये बाकी ये तीनों, तो ये इनका ड्रग का मेटाबॉलिज्म कम हो जाएगा और इनका ब्लड में लेवल बढ़ जाएगा और इनका टॉक्सिसिटी, ठीक है। जैसे यहाँ पे प्रीवियस चार्ट था, अगर बार्बिट्यूरेट लेंगे उसके साथ में ये ड्रग लेंगे, तो बार्बिट्यूरेट का एक टेंडेंसी है, इट इज एंजाइम इंड्यूसर, वो मेटाबॉलिज्म जल्दी करेगा बैल में जब, तो उसके साथ में वो बैल इसको ही नहीं, इसको भी मारेगा, ये वो बैल बार्बिट्यूरेट को नहीं, ये बाकी ड्रग को भी मारेगा, मतलब इनका भी मेटाबॉलिज्म कर देगा जल्दी, तो इनका इफेक्टिवनेस कम हो जाए। थैंक यू, इट वाज द टुडे सेशन, दिस वाज द एंड ऑफ टुडे सेशन। इन द टुडे सेशन वी हैव सीन all the route of administration, then clinical trials and then metabolism.